So thank you very much for joining in first place. And today I would like to talk about the StreamFind project, which is a data processing workflow designer that we are doing in cooperation with Forschungszentrums Informatik from Karlsruhe, and this is a BMBF funded project. And to start my talk, I would like actually to highlight some motivational aspects, and I would like to highlight also the sources of pollution that we have around us and how much we don't know yet about it. And the reason for that is because we still need some aspects of the topic. One of them is more and easier access to data, higher competencies to understand the data, and with that simple software to manage and process the data, and of course that will result in better opportunities to exchange results so that we are more aware of the surroundings and how much exposure we have to pollution, for example. In a more practical example, if you look at the wastewater treatment plant, and here I have an example of two treatment steps, aerated biological treatment followed by a clarifier. And when I think of monitoring this treatment system, I think of chemical analysis, but also biological analysis, as well as sensor analysis to control oxygen, temperature. And once you get the data from all of these monitoring inputs, let's say, you also see that there's a very large diversity of data coming out from it that we need to handle differently. And of course, this for the user, for the researcher or technician that needs to process and to get the results from this data, that means using a lot of software with very high complexity. So we would like to tackle that. And therefore we design and we set the StreamFind project, and the StreamFind, the basic goal is to actually use open source software, so maximize the use of open source software, open source algorithms, as well as native developed algorithms, of course, so that more easy users can have the power of the community to understand the data, to process the data, and to have better results from the data. As I said, this is a cooperation project with Forschungszentrums Informatik, Karlsruhe. Is funded by BMBF, and also for the project itself we gathered a network of partners which are either users, potential users of the tool, developers itself, but also companies, institutions that can help us deploy the software. I would like to continue with the concept, and as I said, we have different types of data. In this case, we give examples of DNA, mass spectrometry, elements, if you like to look into the elemental composition of a given sample, but also sensors or spectroscopy. The concept itself is to build a platform where we fill it with data processing engines. So this is a metaphor, of course. But within these engines, then the data is processed according to workflows. The core itself contains methods that are similar across the different engines, which is more data dedicated. And the idea is then we can have a similar interface towards the different engines, so that it's more easy for the user to actually jump from one engine to the other, meaning from one data type to the other. And I will give more precise examples to that later. So the workflow within the engine is assembled by methods which are connected to specific algorithms, and these algorithms again is natively developed or using open source software, and here I give some examples of the external software that we are using. And this is, of course, an ongoing process, that integration, and the workflow is assembled via processing settings, which is like instructions to assemble the workflow, and I will go more in detail later about it. On the other hand, you have then users which use these processing settings to assemble the workflow, and then from the engine they get the results. Not only visual results like plots, but also the actual data that is being processed. And the interesting thing and an aspect of relevance is that we would like to stimulate this feedback loop between users and developers. So often you see open source software being deployed on GitHub as a repositories or just lost in the huge scientific literature we have, with little attention, and would like to highlight these processing steps, highlight these capabilities, and then to actually engage the users to know where it came from, and potentially give a feedback for improvement, or of course also use the algorithm itself better. And I will describe a little bit more how we do that later. To come back to this wastewater treatment example, when we assess the wastewater treatment performance, of course, then these engines are used for the different data types, so that you have the users have a more unified and easy to access interface, so that it's also more easy to correlate different data types together for more advanced analysis. That's also the focus of allowing more advanced application of data processing tools and algorithms, of course. Right, going to the specifics and how that is used. So this presentation was actually built with a personalized template for I/O slides using R Markdown in the background, meaning that the code you see here was actually run in the background. So this is actually how you would use the StreamFind. StreamFind is essentially an R package with using external software with other languages and some other, of course, algorithms also developed natively in R. But you can use it as a simple R package, and as a scripting language you can build your script, as I will exemplify here, and then have your workflow run as a script that you can always source and process the data. So a little bit about the lines. So the first line is just creating this engine. So in this case is for mass spectrometry, it's called mass spec engine, where you have like the method new. The files is an argument, and this is just a location of mass spectrometry files on disk where we can parse the data. We then create the engine, and then we have methods that are accessed via the $sign, where we can get the number of analysis, for example, or plot the base peak chromatogram, or just get the raw data, so the data behind the plot. That is something that we always had. If you have a plotting method, you also have the get method, you just get the data. And of course this is interactive, because we are using, again, open source tools, so we are using Plotly, that can give this interactive feel of inspecting the data. So that's already there. And once you have then the engine created, the most important aspect of StreamFind is then assembly the workflow. And here you have an example of a basic workflow for non-target screening using mass spectrometry. I will not go in detail into the actual steps itself, but I would just like to give a perspective of how many diversity in terms of software that we can use for this workflow. And here we see not only different software itself, but also algorithms implemented from literature and so on. These are the ones that I kind of use in a more routine basis for non-target screening. And this gets then, let's say, integrated in the engine, and that is of course coming from the developers, where we create the interface to the engine, we create these processing settings, and the only interface for the user is, again, the data that is added, the instructions to build the workflow, and then the results coming out from it. Meaning that these processing settings, initially we spend quite some time on developing them, like what is the structure that we need to really allow the consistency of integrating a new algorithm, but also the consistency of applying different processing steps the same way. So ultimately it's a class where we always add, of course, the processing method name, the algorithm that is being used, and that's because we have different algorithms for a given method. For the same method, different algorithms, and that is because sometimes one algorithm is better for this scenario in comparison to the other one. So we can have different algorithms for one simple task, and we add that information, as well as, of course, the information about the developer or link to other documentation. And ultimately, but not least, of course, is a list of parameters which are conditions to apply the algorithm. As an example, I give here the centroid spectrum method, which uses the algorithm QCentroids from the Q algorithms software, which is then developed by Gerrit Renner in his group in the University of Essen. And this is already published, and you can have a look at the publication. And we have integrated these as a method into the mass spec engine for centroiding mass spec from profile to centroid data. So once you have the settings, The way that you can add it to the engine is add settings. So it is a list of settings which can be like a JSON string, as I see here, give here as an example. You add these to the agent as settings, and then you can then build the workflow. So the order of the settings that are added are directly the workflow, and then you can print from the engine. So once the workflow is optimized, so the processing sets are optimized, you can just replicate that with the function run workflow. One aspect of these processing settings being used as instructions is that you can then save it as a JSON and then reuse it for new data, or just share it with others for better inter-laboratory comparisons, for example, where people are sure that they are using the same conditions for processing the data. It is vendor independent, meaning that even if the other laboratory is using different MS system, this is vendor independent, so that the data can be compared because the processing is done the same way when it is possible to use the same settings. Yeah, and then if you notice, the last step of our workflow was suspect screening. So we were identifying chemicals, and the data behind this is MS data from influent and effluent of an ozonation treatment step for wastewater. And we can see here like a quick overview of the chemicals that we identified. For example, candazartan was identified in both positive and negative ionization. And we can see here then the relative intensity. So we see that in influent is relatively higher, as in effluent, as expected, that is degraded during the treatment, but not completely. So again, the data behind the plot can be just accessed with GET methods. Switching from mass spectrometry to Raman spectroscopy, here we create an engine for handling Raman files, and as you can see, the interface is pretty much similar. So that's what we would like to achieve with this core engine that replicates the interface across different data types. We plot the spectra or get the data. Also, workflow for processing Raman data can be assembled the same way. So use the processing settings, print workflow, and then we just run it. And here I have an example of the monoclonal antibody bevacizumab with where it's present in different concentrations, and then you can see that actually the raw spectra looks like this, and after processing you have like a spectra where you can make a better comparison between different samples. So between the 4 and 5 it's pretty similar, but when you look at the first one where the concentration of bevacizumab was way lower, you see that there's a lot of noise, but the main bands are still there, so you can still identify the bevacizumab after processing in this sample. Going further and also focusing on mass spectrometry, but a completely different approach for workflow is where the workflow implemented for the convolution of large molecules in MS data. So here you have an example of bovine seroalbumin, which is a protein, where we measured in replicate, so 12 files for triplicate for 4 different BSA products. And then here we plot the spectra of the 3 of them, and these different peaks, of course, is then the multi-charge spectra. The workflow itself, of course, is then the convoluting the multi-charge, calculating the actual mass, so that we can more easily compare the different products of BSA. Right. So expansion of the StreamFind is focusing on creating new engines, of course, with other approaches for other data types. And of course, expanding the functionalities of the engine itself by creating methods, and that of course comes from open source community. And here you have some examples of current activities we have going on. And finally, the big picture. So we focus on StreamFind, and that's what I presented, but we are creating StreamPort, developed under Python, and that's because, for example, for sensors, it has a better interface. For machine learning, it has scikit-learn, which is a library with already a lot of assets that we would like to use. And StreamCraft is developed in C++, and that is already something that is supporting StreamFind because it's faster. And we are using for reading data. Yeah. So the idea again is that we can So my voice is a bit gone. But I'll continue. So the idea is that we can impact different fields of activity. So wastewater, drinking water, but also healthcare with quality control of pharmaceuticals and so on. So thank you very much for your attention. So questions very welcome. And again, you can find more information in the ODA project StreamFind with this link. Thank you.
Yeah, we thank